Welcome to our Talks with Tutors. Today I'm very pleased to introduce Professor Rob Gilbert, who's a fellow here at Magdalen College and teaches in biochemistry. He's going to provide a talk this afternoon for you, about 40 minutes. And then at the end we'll have about 20 minutes time for questions and answers. Please feel free to either turn your mic on and speak if you'd like to, or pop it in the chat, and we'll be able to answer those at the end. You're also welcome, of course, to ask any questions more generally about studying at Oxford, etc., or talks about the presentation itself. I'll hand over to you, Rob.
Thanks very much indeed, Amy. Thank you. What I'm going to do now is I'm just going to share my screen, inevitably. So I'm going to talk to you today on this topic about, as it says, evolutionary conserved mechanisms in infection and immunity. And what I'm going to tell you about is some work that I've been involved with since I did my PhD, which I did at Leicester University in the 1990s. And then I moved to Oxford in 1999, and I've been here ever since. And during that period, I've continued to have a particular interest in a family of proteins that when I was working on them during my PhD in Leicester, we just understood to be bacterial toxins, which the bacteria use to attack us and to cause infection in us and ultimately to cause disease, but which we now understand to be a family of proteins which you find in every kind of cellular life on the planet. And the basic idea with these proteins, these pore-forming proteins, is that they start out as a series of soluble monomers, which are in solution, and those soluble monomers will then bind to the surface of a membrane. Here you can see my membrane cartoon here, and as you'll know, it's got a lipid bilayer which constructs that membrane, which you have your positively charged or negatively charged, but charged head groups of your lipids, and then your hydrophobic lipid tails, which are packing together to form the bilayer that structures this membrane. And these soluble subunits of the protein have this astonishing ability first to assemble on the membrane into a ring structure or different kinds of structures, and we'll talk about the different structures they can form in this talk. And then having assembled into a ring, they can then punch their way into the membrane to form a hole. And as you can imagine, if a bacterium does this to one of your cells, what's going to happen is that having damaged the outside of your cell, it might allow the bacterium to gain entry into your cell. Once they're inside your cell, maybe they were consumed by one of your cells, like a macrophage in the blood, which goes around hoovering up bacteria and other invading entities and debris and things. It can then, if they're inside the cell, they can then escape from the lipid package, the vacuole or endosome that they were taken into the cell within, and then grow in the cell. But that process of starting out as a soluble monomer, load of soluble monomers binding to the cell surface, and then punching their way into the membrane is the critical model we're talking about. And these proteins were discovered really by people obviously identifying organisms which cause disease. So these are some electron microscope images on the left here, and this is what's known as an atomic force microscopy image. It's a form of microscopy which gives a closer up image of the structure of molecular systems, particularly looking at their surface, the shape of their surface. So you've got this atomic force microscopy image here and these electron microscopy images here of examples of pores formed by some bacterial toxins, some bacterial pore-forming proteins or pore-forming toxins. And those toxins were identified, they were discovered by people knowing of organisms which were causing disease, such as Streptococcus pneumoniae or the pneumococcus, which produces this toxin, pneumolysin, or Streptococcus pyogenes, which produces this toxin, streptolysin, or Clostridium perfringens, which is the cause of gangrene, one of the causes of gangrene, that produces perfringolysin, and discovering that these bacteria seem to have the capacity to, well, actually, what they found was it had the capacity to lyse red blood cells. Now, the lysing of red blood cells isn't really what's going on That's not really why these organisms make these toxins, but it's a good way for us to assess what their activity is, or to find that these bacteria make proteins which have pore-forming activity, which can damage our cells. And one of the ways in which they were discovered then was to, first of all, identify that these organisms, like the pneumococcus and Staphylococcus pyogenes and Clostridium perfringens, that they were producing something which caused red blood cells to lyse, and then to try and identify what it was that they were producing that was causing the red blood cells to lyse. And one of the ways they identified what it was that was damaging the cells, and which they then could say, well, maybe this is allowing the bacterium to infect us and invade our tissues and so on. One of the ways they did that was by imaging it and finding that there was something that the bacteria that were producing that produced assemblies you could image using electron microscopy, as you can see here in these images. You can see in these images, let's look at this middle one because it's the easiest one to see. What you can see here is you can see dark holes here, and each of these dark holes is a hole that's been made through a membrane. And you can see then a kind of lighter patch. This has been negatively stained, that's why it looks lighter. This lighter patch here, which is the protein sitting on the surface of this cell membrane. So what's happened is that, as you saw in the previous slide, the monomers of this protein, a load of copies, have bound to the cell surface, assembled themselves into a structure, and inserted in to form a pore. One of the things you're going to notice here, which is interesting, and which is one of the particular areas I've worked on, is that these are not complete rings. They are arc structures, so they're not actually a bit broken. And it would be possible to think that maybe they had been complete rings, but they broke up. But actually, the fact that the membrane all around is still complete suggests that these really are structures in which you've got a protein bit here, which is inserted to form a pore, but you also have parts of the membrane which are helping to complete the pore. And while it is true that these systems form pores just like we saw in the previous slide using rings of subunits, they also form pores using these broken structures too. And as you'll see, that turns out to be quite important in the way they work. But I already said that this was a very widely spread family of proteins, and later on it turned out that, for example, there were other proteins that were being made. This is from Metridium cinele, which is a sea anemone. So metridiolysine from the sea anemone. Here, this forms similar kinds of structures on membranes. And this is perforin. Now, this is perforin. This is a human protein or mammalian protein. And our white blood cells that are known as killer cells or cytotoxic cells that go around, for example, eliminating cells in our own bodies that are infected with virus or which are showing signs of transformation to potentially become cancerous. Perforin, which we make to do that, this perforin, it also forms similar kinds of structures. But interestingly, at the time when people were starting to image metridiolysine or perforin or other proteins that they were identifying from mammals and other organisms that seem to damage cells by forming pores, interestingly, they didn't make the link that maybe these proteins and these proteins were all related. They for some reason, in retrospect, it was a bit odd that we didn't really think about them being related, but we treated them as if they were two different families. We had this family that were bacterial and this family that you find in eukaryotes, like the sea anemone and human beings. So in the time that I was doing my PhD, people really were focusing on the bacterial members of the family, and one of them that they focused on was perfringolysin, which you see two images of here, one from electron microscopy and one from atomic force microscopy. And you'll see some more atomic force microscopy in the main research part of my talk after I've introduced what I want to talk about, because I'll show you some movies from atomic force microscopy so you can see real time, close up, what these proteins look like when they're doing what they need to do. So looking at the bacterial members of the family, one thing that happened was that a group in Australia solved the atomic structure of one of them, which is called perfringolysin, here, this one. The perfringolysin structure, which they solved by X-ray crystallography, showed us a very interesting structure, and many of you will have seen protein structures in studies that you've done at school already. And as you can see, this structure consists of, it's got beta sheet structures in them, which, as you know, is one of the main types of structural motif or secondary structure that you find. And the other main one is alpha helices. You can see it's got alpha helices here. And I've colored here a set of alpha helices in red. And the reason why they're colored red, you're going to see in one moment. So when I was doing my PhD, a group in Australia solved this structure. And then during my PhD, what I did was that I took another one of these family members, pneumolysin here, and I purified it. And then having purified it, I then got it to assemble structures on membranes, on synthetic membranes that we had in the lab. And then we imaged them again using electron microscopy. And then what happened was that because electron microscopy at this stage had become capable of not just giving these 2D images, but by freezing our samples without any stain, and again, you'll see an example of the kind of data we use for that these days later on in the talk. By freezing the sample, we're able to image it in such a way that we can actually see the molecular details themselves of the actual molecules on the membrane. And then we can take, if we take many images of objects that we're looking at by electron microscopy, we can reconstruct them in 3D, as you can see here. And what we did was that we found two different structures that we could see. One was this structure here, which has, as you can see, a kind of, it's like a sort of soup plate appearance, and it's got this sort of ring on top, and the membrane is complete inside. You can see this side view. You've got these bits of the protein sticking up from the surface, and here's the membrane complete inside. You can see the lipid bilayer there. And then what happens is, what we discovered was that it then inserts, as you saw in my cartoon, it hole punches into the membrane to form this pore. And having done that, you can see now the lipid's gone from the middle of the membrane. And so now you've burst a hole in the membrane. And having put that hole in the membrane, that's then going to allow the contents of the cell to leak out, or the cell to become inflamed, or the bacterium to infect the cell, or different things that might happen depending on the evolutionary reason why the protein was evolved by the bacterium. And if we model this structure up here that was solved by crystallography into our electron microscopy maps, we can build a model up which tells us what the whole structure looks like when it's formed, this what we call a pre-pore structure. This is not quite formed a pore, it's just assembled on the membrane, and then when it's formed a pore. And what's really amazing is that we found, and others found too, using other kinds of methods, that it goes from this structure here to this structure where these alpha helices have refolded into beta sheets. Now, when we learn about alpha helices and beta sheets, we usually think of them as being separate kinds of things. You either form an alpha helix or a beta sheet in a protein. But actually, in this case, what we found was that the alpha helices could convert it to beta sheets. And that reminds us that the structure that proteins have depends on the environment they're sitting in. And so when this protein is in solution, it's happy to have these helical structures as its stable state. But when it gets inserted into the membrane, when it's forced to insert into the membrane by assembling on the membrane surface and then punching through it, when it does that, that region of helix refolds into beta sheet. It was an amazing discovery at the time, and something that is really kind of characteristic of this huge family of pore-forming proteins. Just here's a little bit more of historical images of these proteins. This is quite an old image, very old image now. This is actually the first image discovering something called complement. And complement you may have heard of. It's part of, it's in our serum, it's in our blood, and it's one of our innate immune defenses against bacterial infection. And you'll see something more on complement a bit later. So these are images of what's known as the membrane attack complex. We make this to target bacteria, especially bacteria from the Neisseria family, which includes Neisseria meningitis, which is one of the causes of meningitis. And so here you can see some images of complement. And here actually are some images which are from when perforin was being discovered, or I should call it now perforin 1, because I'm going to introduce you to perforin 2 in the main research part of this talk. So this is one of the images from when perforin was discovered, and you'll remember that perforin, or perforin 1, is a protein that we make in our white blood cells to target other cells of ours, to get rid of them, to kill them. And we do that because those cells that we have are recognized as being infected with a virus, or because they are showing signs of being damaged, such as to potentially form cancer. And down here you can see yet more images of perforin and some more atomic force microscopy images. And what you can see is, again, you've got a mixture of complete rings and broken structures. And we can even look at the function of these using measurements of the electrical conductance through membranes. And you can see here very nicely an example of how you have two of these arc structures coming together to form a pore, and there's lots of current going through there. The pore falls apart spontaneously, and then it forms this kind of structure. It's much noisier, and that's because the structure includes lipid, and you can imagine the lipids are moving around when the protein is actually forming quite a solid structure, and then they come back together and form this solid state again. So we can see dynamically these systems assembling and disassembling as they damage the membranes to which they're bound. So we've got to remember that actually that there are kind of variations on this hole punch theme. There's this variation here, which is that you can sometimes get pores formed not by full rings, but by arcs of subunits. And another variation on the theme is that this is actually the complement system, the membrane attack complex, and actually that doesn't just form from one protein, it forms from these proteins, C5b to C8, coming together. They all come together to form a kind of a nucleus, a nucleating complex, to which this C9 then forms the rest of the pore. And so there's different kind of variations, but what's common is this idea of going from a monomer into a membrane inserted form and changing structure from an alpha helical to a beta sheet structure. And this here down to show you a little bit more detail about complement. This is now an atomic structure of this system that I've got in my cartoon here. This is an atomic structure of it, which has been studied by crystallography and electron microscopy now. So we have a full picture of how the membrane attack complex works on membranes. This is another example of the kind of ways we can study these proteins, and it just gives you another idea of how they assemble. So on the left here, this is a real-time movie of a fluorescently labeled version, in fact, of the perfringilicin we were looking at before. And what you can see is that there are various dots which are getting brighter and brighter, and why these are getting dots, you can see in the background there's lots and lots of fluorescently labeled protein racing around. And then what's happening is that the dots are getting brighter, and they're getting brighter because they're assembling into these supercomplexes. So we can follow them as they assemble on the membrane, see in real time how they assemble, and then we can actually combine this even with measurements of their conductance through the membrane to relate to each other the idea of the proteins coming together into this ring or arc-shaped complex and the proteins making a whole. But you will see that the objects that you see are of different sizes, and you know that because they're of different brightnesses. So some of them are brighter than others, and that's because some of them have more fluorescently labeled copies in them than others. And just over here, hopefully this should, yes, just over here, what you've got examples of a close-up. These are two smaller complexes coming together to form a larger complex. There we go. And then down here, I'll just run that again. Down here, we've got two that were together and have now gone apart. So, and then these are two are coming together. And we can use this to follow in real time the assembly of our protein. So we can get quite a good understanding about how the proteins assemble and how they come together to form these complexes which attack membranes. Now, as I said at the beginning, when I was doing my PhD, people really thought of these pore-forming proteins in two different groups. And perfringilicin is a protein you've met before. It's the one that we've been talking about, and you just saw some images of it racing around on a model membrane surface, forming pore complexes. But about 13, 14 years ago, people first solved the structure of examples of members of another family of proteins, the family that perforin belongs to. So we have the bacterial proteins here, which were known as cholesterol-dependent cytolysins because they bind cholesterol in membranes in order to target the membranes and attack the cell. And then there were these perforin proteins, of which perforin itself, perforin 1 is an example, but other examples, this is actually from a microorganism called Photorhabdus luminescens, and that makes its own version. And then this is actually another one from mammals, actually. This is one that we make, which is involved in cell guidance in the brain, in the cerebellum. But when these structures were solved, and first of all, this was one of the first ones to be solved, and the first two to be solved, you can see the way it's colored here, that they actually have very similar structures. So you can see very clearly, this has got this structure with this dominated by this beta sheet here, and then with these alpha helices either side, which you've seen form beta strands when it forms a pore. And then here you've got the Photorhabdus luminescens one, and that's also got this beta sheet with these alpha helices. You see, there are differences, but the structure is really very similar. And there are all sorts of reasons why we need to believe, and there are all sorts of reasons to believe that these are examples of divergent evolution. So there's common ancestors to these proteins, even though they're in very different organisms. Now these are in two microorganisms. This is in Photorhabdus. This is in Clostridium perfringens. But this is in mammals, okay? And this is a very closely conserved protein found in all vertebrates. This thing called astrotactin, which is involved in brain development. And then this is perforin 1, which is, as I say, involved in cytotoxic cell activity against our own cells in what we know as immune surveillance, checking for damaged cells in our bodies. And if you look at the amino acid sequence in these proteins, you find that they really are very, very different from each other. In fact, the reason why we didn't know perforin 1, for example, was related to perfringilicin was because you can't find any sequence similarity between them if you just look at the sequence. If you just compare the sequences, they're far too different from each other for you to find any reason why you might think they're related. And what we can do is we can do a statistical analysis of some regions of the sequence which seem to be characteristic of these proteins. And you can see two of those regions here. One is called what we call the MACPF signature motif, or the membrane attack complex perforin signature motif, and that's labeled up here in the middle of this beta strand. And there's another region here, which is actually a critical region at a hinge point in the structure, which is called this GGXW motif. W is the residue tryptophan, G is the residue glycine, amino acid glycine, H is histidine, and so on. There are different amino acids here represented by their single letter symbols. So if you were just to compare these sequences, you would find no similarity by looking just pairwise at the sequences, because we have the structures. We can see that perforin 1 and perforin lysine are in fact related to each other. And having discovered that, we can then look at how their sequences align, because we can align their structures, and then having aligned their structures, we can look at aligning their sequences, and then we can see where the sequences match. And what you can see is that there's very limited agreement. In fact, there are just two glycines, which some of you will know is the smallest amino acid. It's the simplest amino acid, in which the R group, the group that makes amino acids different from each other, is just a proton. So these two glycines are the only two residues common to all members of this family. All the other residues vary completely. And what is conserved here is the structure, and the sequence has just varied through billions of years of evolution. So you can't detect the relationship between perforin 1 and perforin lysine without solving their atomic structures. But now, with more sequence data, and as you know, many organisms are now being sequenced, with more sequence data, and in hindsight, with knowledge of the structural comparisons, we can start to construct sequence-based phylogenies, or evolutionary trees of relationship, which describe the relationships between all these organisms in terms of sequence. And this is, as far as I know, the most complete study that's been done of this, not by us, but by a group in the States. And what this does is it clusters all the different sequences of all the different proteins into a set of families which are more closely related to each other than they are to the other sequences in the data set. So you've got hundreds of sequences, and what you do is you try and work out which sequences are more closely related to each other than they are to other parts of the data set. So one very well-defined group in the bacterial form, we've already met these. These are the ones like perforin lysine and pneumolysine, the cholesterol-dependent cytolysins. And you can see that they here, sitting at this branch on the evolutionary tree, they here reach back down here to, this is the kind of theoretical origin of all life. This is the theoretical last universal common ancestor of all these organisms that make these proteins. So here they're the bacterial ones. And then you've got some others. Let's go to the top of the tree, as it were, or the most developed part of the tree over here and over here. Now, this is where you might find these are some eukaryotic forms. This is perforin 2, which I'm going to introduce you to shortly. This is perforin 1 we've been talking about, which is involved in our cytotoxic T cell response. This is where you find the membrane attack complex proteins, so the complement proteins. But you can see that these proteins are found all over the place, as I said, in fungi, in plants, in insects, in sea anemones like Cnidaria and so on, and even in things like parasites, like the parasites which cause cause malaria, Plasmodium and Toxoplasma, two examples of a family of apicomplexan parasites. We're going to meet the role of the proteins in those organisms in a minute. So you've got this amazing breadth of spread of these proteins all over evolution, all over the biosphere, in every form of cellular life. And what I have done here is I've just highlighted a couple of relationships for particularly important proteins. So here's perforin 1, very important protein. It's a protein which is, as I say, involved in immune surveillance. It helps to protect you from cancer. It helps to protect you from viral infection. And if you follow the evolutionary tree back, you can see it branches out a couple of times and comes back down here, very, very relatively circuitous route to the origin of all life. Here's complement over here. And so what I can tell from this tree, actually, I can see that perforin 1 in our cytotoxic T cells and complement in our blood, which is floating around in our blood as part of our innate immune system, they are kind of similar age to each other evolutionarily, and they share a common ancestor here. Here, if I come back here, you can see, for example, here are these ones that you find in parasites like Plasmodium that causes malaria. And you can see that has a common branching point here, where it split off, that family of proteins split off from this family of proteins involved in immunity for us here. But I just want to target here momentarily perforin 2, because if you look at perforin 2, it's a very interesting protein. Perforin 2 is found in every form of animal life. So every animal, from sponges to humans, the most simple animal, sponges to humans, has perforin 2. And if you look at its evolutionary relationship, you can see if you follow its pathway back, it's very close to the universal ancestor of, in fact, of all life. And in fact, we believe it's the closest known perforin family member to the origin of the whole family. It's an example of something that's closest to the whole family origins. And you can see that that's closely related to, for example, a form of protein like torso-like, which is made in insects. So there's actually some relationship here between some aspects of our development and our immunity, because perforin 2 is involved in both development and immunity in humans, and also development and immunity in other organisms like insects. So, well, I'm now going to go, and you'll see this slide again shortly, because it gives a sort of global picture of all this family of proteins that we're talking about. But I now just want to look at one or two examples of how these proteins act. So this is Toxoplasma Gondii. It's a parasite which causes disease, particularly in the immunocompromised, for example. And they produce a perforin-like protein, which we've done some work on. And this is from somebody we've worked with in the past. And you can see that what this protein does is that it enables these tachyzoites from Toxoplasma, these little single-cell parasites, to break out of our cells. And we can actually do an experiment where we stop that protein working. So if I just do this, I'll just do. What you should be able to see now is you should be able to see on this slide how the parasites break out of these cells here. There we go. And then if we look on this slide here, this is if we knock out the protein. So it does get to burst out there by sheer force, but here you can see the parasites can't get out. So that goes to show that this protein is really critical in the ways in which the parasites break out of cells to cause disease. And here's some other examples. So this is from the malarial causing parasite. This is the cell form called the sporozoite from the Plasmodium that causes malaria, swimming around, you can see, very fast. If we knock out that protein, the perforin-like protein 1, then it can't really do anything. You see, this one's trying to get into the cell, but it can't get in. And here's an example again of wild-type activity. This is the parasite moving inside a liver. So it can't do that to invade our tissues if it doesn't have this perforin-like protein. So what I've shown you is that there's a family of proteins, and I've shown you just a small number of examples of that family, which is known formally as the membrane attack complex perforin cholesterol-dependent cytolysate family, because to reflect the dual origin of the history of our work on these proteins, that they're found both in mammals and other organisms, other animals, and they're found in bacteria, these cholesterol-dependent cytolysins. But they're everywhere, and they're involved in every kind of cellular life. They're even found in some viruses. So we have the membrane attack complex perforin, or MACPF family, which is in eukaryotes mostly, and the cholesterol-dependent cytolysin family, mostly in bacteria, mostly gram-positive bacteria. But they're actually the same family. We only discovered that by looking at their atomic structures. And the basic understanding we have of the way they work is that they do form these pores. As you've seen, they form these. This is an electron microscopy study we did about 5 years ago, published about 5 years ago. What you can see here is this is the prepore structure. It's an arc of subunits on a membrane, and then it inserts to form a pore. And some people will ask, well, why is that a stable structure? Because here you've got the protein inserted, but here's the membrane. Isn't the membrane just going to fill in the gap? And actually, it turns out the lipids can reorganize themselves to form a structure in which they do form a stable pore with a well-defined barrier between the water here, which is going to allow leakage from the cell that's been attacked, and the membrane that's closing the gap on that open arc of subunits. And the reason why this is useful, this is where we segue to thinking about the research case I just want to present to you briefly. The reason why this is useful is if you look at the example of listeria-lysin from the pathogen Listeria monocytogenes, again a pathogen of immunocompromised people. If you look at that, you discover that it forms what we call patchwork pores, in which these kind of incomplete assemblies come together to form larger assemblies, and these actually turn out to be important in how the bacterium grows and infects and grows and moves from cell to cell. And my feeling is, my own view is that the reason why this protein fold has been so conserved throughout the whole history of cellular life, from the last universal common ancestor of Streptococcus pneumoniae and human beings, we've all had these proteins. And the reason why is because they have this particular trick of being able to form pores in which the more subunits you have, the larger the pore is you get. So it doesn't have a defined size, and that means you can form different sizes of assembly to do different things at different stages of a life cycle, at different stages of an infection, for example, too. And this is where we think in detail about one particular protein we're going to talk to you about, which is perforin 2. But to get there, I'm going to talk about Listeria a bit more, because we've already said Listeria, it's a pathogen, it's an intracellular pathogen, so it invades our cells. When it invades our cells, it forms pores in the endosomal membranes, the membranes of the vacuoles into which it goes, and it forms these pores in order to stop those becoming acidified, because we try to acidify them to kill the bacterium. And that means the bacterium is able to grow inside them. And then what it does is it forms some larger pores. As you've just seen, it can form smaller pores or larger pores, depending on how many subunits it puts together. And having done that, it can then escape through those pores to then, swim through the cytoplasm and then invade the neighboring cell. But we make a protein called perforin 2. And this perforin 2 is present in macrophages, which are cells which go around hoovering up, as we said, debris and bacteria and other potentially invading organisms like parasites from your blood. The macrophages go around and they're hoovering up the debris to recycle them for your body's own use and for your body's own protection. But in fact, perforin 2 is inducible in any cell that gets infected. Any cell that gets infected can make this perforin 2. And this perforin 2 is made in our cells as it's a membrane-inserted protein. So unlike many of the other proteins I've been talking to you about, which are actually, they're made as soluble proteins which then refold, this has a soluble domain, but it's tethered to the membrane. What happens is that when this becomes active, we believe that this linker between the soluble region of the protein here, consisting of this perforin-like domain or MACPF domain, and this P2 domain, which is characteristic of perforin 2, you'll see the structure of that domain shortly, this region gets cut off from the transmembrane region, and then it can target the bacteria that are living inside your cells. And what happens is, targeting the bacteria, it kills them. So basically, by mechanisms we're not entirely sure about yet, but we're working towards them, and I'm going to show you how we've worked towards them so far, this protein will target the bacteria. But what is the point here, of course, is that here we have an amazing situation where we are using a protein which is a member of this family of perforin-like proteins to attack the bacterium, and at the same time the bacterium is using a member of the same family to attack us. And this goes for any number of other parasites or organisms that are making these perforin-like proteins to attack us. We are using our own proteins of the same family to attack them, and that means this must be a very important family of proteins to be used kind of for the same mechanical reason, or the same specific reason to target a membrane, but by organisms who are struggling with one another to either infect or to protect from infection with respect to each other. So we wanted to study perforin 2 and understand how it does what it does. And so what we did was that we purified the protein, and what we could do is we could get it to assemble into these ring structures by lowering the pH to 5.5. And what you just saw there was you saw some atomic force microscopy, and I'll show it again. You saw some atomic force microscopy here of the assemblies sitting on a surface that they're imaged on by atomic force microscopy, and I can't get it to work again, I'm sorry. But by taking these electron microscopy images, what we can do is we can then classify them together, and classifying them together by finding similar images, we can average them, and by averaging them together, similar images, we get different views. You can see a very clear view. You can see, if you counted, you can see it consists of 16 subunits. get some views which are incomplete, but you get many different views, different tilts, different views, different viewpoints of this assembly. And having taken all these different views, what we can do is we can then take the different views and combine them together to form a 3D map. And here you can see a 3D map, a 3D electron density map formed using electron microscopy. And you can see the surface of this map, and it's just contoured, it's just colored to reflect how well defined the map is, what the resolution of the map is. But that map is good enough for us to see the atomic side chains of amino acids, because it's at resolution at about 3 × 10⁻⁻10 meters, or 3 angstroms. And you can see some of the examples of electron density we get here. Here's some side chain density, and you can see these bits of the amino acid side chain sticking out into the density of our protein. So it's a well-defined structure. So what does the structure look like? It looks like this. So you've seen it has 16 subunits, and it forms this sort of dish structure again, as you've seen before in cartoons. And it consists of these subunits, and they're kind of folded against each other. And one really interesting way in which they're folded against each other is that this orange bit here is what we call the C-terminal tail. And the C-terminal tail is kind of folded back against the next door subunit. So you've got 2 subunits here next door to each other, one copy here, another copy here. And this orange bit of this second copy is folded back on the copy before to strengthen the whole structure. And that's one of the important ways in which this structure is stabilized. But this structure poses a big question for us, because we can work out by various means, we can work out that actually this region here at the end of this P2 domain, this beta-stranded region, is the region where it targets the membrane surface. But the reason why it's confusing to us is that that's poking downwards there towards the bottom of the slide, but these helical regions, which we know are involved in membrane attack, are poking upwards in the other direction. So it looks like the attack bit of our protein is facing the wrong way from our membrane binding bit, and that's different from all the other members of the family. In all the other members of the family, this bit here and this bit here, the pore-forming regions of the attack domain, would be facing the same direction. And we were trying to publish this. The reviewers of our paper said, oh, couldn't this C-terminal tail be important in binding too? So we deleted that. And what these studies show is actually that it's not important at all. So it really is this bit of the structure here that's important. So we have a big problem here. We have the attack bit facing that way and the membrane binding bit facing that way. So how is this attack bit going to be able to attack this bacterial membrane when it's bound to it, when it's facing the wrong direction? Now, I just want to point out to you again that this perforin 2 is very close to the origin of all this family of proteins. So things we can find out about how this protein works might be relevant to how we understand the whole family to exist. And I want to remind you that we can work out using sequences now that we know that these families are related to each other, the bacterial family of proteins and the other proteins. We can use sequences now to compare them. What we can also do, though, is we can build phylogenetic trees or trees of evolutionary relatedness, which are based on the structure alone. And this reminds us, of course, of the famous diagram that Charles Darwin drew when he was thinking through the theory of natural selection. So really, we can map out evolutionary history using the structural studies that we do. By taking the atomic snapshots that we do of the protein folds, we can see how the bacterial ones are down here on one end, and then the mammalian and other members of the family up here on the other end of our evolutionary tree. But we wanted to understand how this protein really does work to form pores in membranes. So what we found out was that if we reduce the pH further, we could get it to form a pore. So now what you can see is, you can see here are some more electron microscopy images. Here's the pre-pore form. You can see it sitting on the membrane, and the membrane is still intact here. And then if we reduce the pH to pH 4, we get a hole forming. You can see the hole forming very clearly here through the membrane. Another thing we can do is we can look at that kind of using a functional assay. And the functional assay we used is to look at fluorescent. We have a fluorescent dye, which has different sizes. This is 4,000 atomic mass units, 10,000 atomic mass units, and 70,000 atomic mass units. And if you see, when we add our perforin 2, if we have the 4,000 one, then you can see that we've got a red dye coloring our membranes, a green dye in the buffer around our synthetic membranes, our vesicles, our giant unilamellar vesicles. And what we find is that without perforin 2, of course, the dye stays outside. We add perforin 2, the dye can get in. If it's the 10,000 atomic mass units, of course, without perforin 2, it can't get in. When we add perforin 2, it can get in, but doesn't equilibrate completely. So the density of the dye inside is different from the density of the dye outside, whereas the densities were the same up here. And then down here, if it's 70,000 atomic mass units, you can see it can't get in. So the pores have a defined size, a limited size. And here you can see some more of this atomic force microscopy. This is real-time imaging of the pore formation. You can see you get a big conformational change. And you can see it because it gets much brighter. It's because it gets closer to the tip surface of the microscope that's looking directly at these set of complexes. And then here you can see more of these pre-pores, and what you can see is them converting into pores, and actually they do it in a clockwise fashion, it turns out. So it turns out they one subunit goes and then they rotate around in a clockwise way to convert to the pore structure. So what does our pore structure look like? So we solve the structure again using electron microscopy. Here again are some images in different views. Here's a top view, here's a side view, and here's a side view down here of our 3D map. So we're able to do that. And having done that, we can then get a sense of what the structure looks like. So it's 16 subunits again. And it forms this beautiful barrel structure here. And we can see how the structure is refolded now, so that you can see that the membrane binding domain and the attack domain are now facing in the same direction when they were facing the opposite direction before. And it forms this beautiful structure which is going to attack the bacterial membrane. So this has involved a massive conformational change. So on the left, you've got the pre-pore assembly of the protein when it's in solution or when it's bound to the membrane first of all. Here's the structure it forms when it's inserted into the membrane. Look at the change it happens. So if we align it just to this P2 domain, the membrane binding domain, it's undergone a 180-degree rotation right around that P2 domain, and a 90-degree rotation actually if you look down from above. And in total, the conformational change is like this. So it's a huge change in structure that the protein undergoes in order to go from this inactivated state when it binds to the bacterial membrane to the active state that's going to kill the bacterium. So we now have a model for understanding how it is that our proteins can target bacterial membranes. What they can do is that inside the phagosome or endosome, the vacuole into which the bacterium has been consumed by the cell that's taken the bacterium inside, or that the bacterium has invaded, if it's Listeria, what's going to happen is you get proteolysis, you get the cleavage of the linkage of the soluble bit of the protein from the membrane anchored bit. And then after cleavage, what happens is it will bind to the bacterial membrane, it'll reassemble, it'll reorient, and because it will then allow it to form a pore like that. So now we have a way of seeing how perforin 2, which is a broad spectrum innate and inducible antibacterial defense that we have in our cells, is able to target bacteria like Listeria lysine by getting into these endosomes, attacking the bacterial membrane surface, and then killing the bacterium. And that's where I will finish. Thank you very much for coming to the talk today. I hope it was interesting, and I hope that it showed you some of the ways in which we are studying the molecular basis for infection and immunity, in this case using the example of a family of proteins which amazingly have been conserved throughout cellular life from the last universal common ancestor of, for example, Streptococcus pneumoniae and any number of other bacteria that make these proteins, and human beings, and which the bacteria use to cause infection and we use to defend ourselves against them.
Fantastic. Thank you so much, Rob. That was brilliant. So now, guys, we've got about 15 minutes for you to submit any questions you might have. So anything at all about what has just been discussed, or as I said, more broadly about coming and studying at Oxford or studying biochemistry. So either pop your hands up and unmute yourselves, or I'll give you a couple of minutes to put something in the chat if you'd like to. But yeah, any questions at all? I guess I can start off with thing to get it going. So I'm thinking if someone is thinking about studying biochemistry or biology or something, what's something they might be looking at doing now while they're still at school before applying?
Yeah. So if everything is really, so what I would say is that one thing we're really looking for always when we're looking for students is, or we're considering applicants, is enthusiasm. And so what you really should be doing is kind of exploring things that interest you, basically. And hopefully there are things that you're learning at school that, I'm sure there are things you're learning at school where you're thinking, oh, that's interesting, I want to follow up a bit more on that. Maybe, well, I talked about protein structure. One of the things you learn about in terms of protein structure is hemoglobin, for example, and how that works. You learn some stuff to do with basic ideas in protein structure, like the secondary structures I talked about, alpha helices and beta sheets and so on. So if you're interested in protein structure, maybe you want to look for a popular science book on the protein structure theme. There's actually a really good one I'd recommend, which is called, it's got a really catchy title. It's called I Wish I'd Made You Angry Earlier. And it's by a man called Max Perutz, who won the Nobel Prize for working out the structure of hemoglobin and therefore how hemoglobin works. But I Wish I'd Made You Angry Earlier is a really good book to introduce you to what biochemistry is like and what it was like, in the 1930s and 1940s and 50s and 60s and so on, when Max Perutz was doing his amazing work, but also what it's like now, because in a way we haven't really taken to doing things differently. We actually just do things maybe a bit more high throughput. It's all a bit more complex, it's more computerized, and some of the techniques are better, but the basic ideas are the same. But yeah, reading around, that's the thing to do, I would suggest. But it doesn't have to be books. I mean, it could be books, and you'll perhaps be aware of different good authors, like Matt Ridley, like Nessa Carey, and others. So and the man who wrote Power, Sex, Suicide, who momentarily I can't remember his name, but I just remember the book about the mitochondrion. But you reading around, but also reading on the internet and just looking at picking up on what interests you. That's the kind of thing. And then when you're thinking about your personal statement, that's what you want to tell us about. What's really interested you, what you've read about. We're not looking for anything in particular. You haven't got to read any particular book. It's just about what you find that really piques your interest and makes you think, oh, that's actually pretty cool. I'd like to spend a few years studying that at university.
Brilliant, thank you. We've had a few questions come through. So Katie says, what careers can come from a biochemistry degree?
The answer is anything, of course. So you can do just about anything. But obviously, so in the biochemical area, research careers is one of the possibilities, and research careers come in different shapes and sizes. So you can do an academic research career. That could be as a researcher in a lab as part of a team. It could also be in the end leading a team in a lab. It could be in industry. These days, there's quite a lot of movement backwards and forwards between academic life and industrial life. So making a choice one way doesn't cut you off from the other at all. So you might go into industry straight after a degree, but you might end up back in academic life later on, or vice versa. People in academic life they establish their own companies, or they form collaboration with companies, or they decide to go and work with companies. So all of that is possible. And that's the kind of thing that usually, almost always, you'd access by doing a PhD, although again, not always. But the fact is, you can actually do almost anything with biochemistry. And I'll tell you, maybe it's useful to give some examples of the kinds of things people I've taught over the last 19 years or so have gone on to do. Those include going off to become a teacher, joining the RAF, joining the army, going to the civil service, becoming a consultant, as in a management consultant, training as a vet, training as a doctor, accountancy, law, all sorts of things. And what's more, also, not all of them have stayed where they first went. So some of my students have got jobs in, say, things like law. I've had a couple of students have got law or sort of consulting, and then they've thought, oh, actually, I'm going to do something else. One of them thought, oh, actually, I'm going to join the army. Another one thought, oh, actually, I'm going to go and do a PhD instead. So he came out of working in law and went off to do a PhD. So there's quite a lot of flexibility. Basically, a biochemistry degree, like any degree, equips you for the future. It gives you a skill set, a way of thinking, a set of transferable skills. Maybe you'll end up using biochemistry yourself. Maybe you'll just be using a basic scientific understanding of the world in another sphere of work. So really, there's a lot of flexibility. And that's one thing about university is go with what interests you, because it doesn't limit choices that much really for the future. And these days, with graduate entry medicine and other kinds of graduate level courses, graduate training in law and so on, it doesn't limit you from going into other things to have done biochemistry. There's lots of flexibility.
Yeah, absolutely. Thank you. So someone else has asked, do you just need chemistry and biology as A-level requirements for biochemistry?
you actually need two sciences, and you need chemistry. You don't have to have done biology. In fact, interestingly, and the reason why we say that is because the fact is that some of the biochemistry we do, quite a lot of the biochemistry we do at university is different from the kind of biology you do at A level. So a lot of the biology you do at A level is more like whole organism stuff, organ function, and that that can be very good preparation for. It's partly good preparation for biochemistry. It's also good preparation for medicine and biomedical sciences and all sorts of other pharmacology, all sorts of other degrees you might do. But you can also get up that kind of information and understanding from reading around and general knowledge. But the chemistry is the thing you really, really need. So most people, as it happens, who apply have got biology, chemistry, and maths. But basically, that isn't a requirement. What you have to have chemistry, and then one other science. And by one other science, we mean maths, physics, or biology, basically.
Brilliant, thank you. We've had a big thank you from the King Solomon Academy, and they've asked whether you recommend any additional reading. So I have made a note of the things that Rob has just said, so I can make sure that's included in an email that goes out. I don't know if you have any to add, Rob, but as Rob said, definitely don't feel like these are what I email out is things that you absolutely should read to apply for biochemistry. Yeah. Only read it if you're interested.
Yeah. And it's just what, like when I applied to biochemistry, so I read biochemistry. I actually read it at Durham. I didn't read it here, but the books I'd read are actually more like around evolution. I'd read Dawkins. I'd read The Selfish Gene. I was really into Stephen Jay Gould, who died a few years ago now, but did some really interesting. He was a lifelong evolutionary biologist. He worked as an evolutionary biologist academic all his life. But he wrote some really good books, which I think really give you an insight into how evolution works. They've got pretty good titles too, like Hen's Teeth and Horse's Toes is a good one. And I read those at school, and those really helped me. Selfish Gene, of course, lots of people have read that. It's a good book to read. But there are lots of books. Don't, as Amy says, don't feel kind of constrained. Go with what is interesting. Go with what you come across. And if in the end what you've come across is interesting articles you found on the internet, fine.
Absolutely. I'm going to come across this notes page in a few months and be absolutely baffled. Just written down. Great titles. Okay, so we've had another question. How different is biochemistry to biomedical sciences?
So what I'd say is that biomedical sciences is more kind of whole organism focused, and it's more sort of the physiology of the system, I would say. Whereas biochemistry does a bit of that. So you do kind of, when you do metabolism, you think about like metabolic disorders, like diabetes, for example, as a key example. But you don't do so much on the whole system. When you think about cell division control, you think about cancer, but it's all about the mechanisms which underlie the cancer. It's not about the development of the cancer physiologically. Whereas in biomedical sciences, it's more about the whole system, thinking about the system, the whole system in terms of its biology. So the molecular details are irrelevant, but their emphasis isn't so much on the molecular, more on the systematic. The other thing is that biomedical sciences includes a lot of neuroscience, and there's no neuroscience basically in biochemistry.
Brilliant, thank you. And then on a similar vein, how would a biochemistry degree be similar or different to a medicine degree?
So in fact, the medicine degree, the preclinical medicine degree, is very like biomedical sciences. They're not entirely the same because there's more infection and immunity in medicine, the preclinical medical degree, and also there's also kind of quite a lot of teaching, obviously, on things like organization of the body for obvious reasons, which the biomedical scientists don't do. So basically, biochemistry again is much more about the kind of principles, the kind of mechanistic, molecular principles of how things work. Medicine is a very practical degree. It's about how things are actually working, and you need to understand something about the molecular basis for that, in order to see enough of the picture to understand it. But you don't need to be studying the kind of the theory, as it were. It's more about the practice. I really like teaching medical students because it's really refreshing contrast between biochemists and medics. They're very different kinds of students, and they're both good groups of students to teach, but they kind of complement one another quite nicely. The medicine is much more practical in terms of how do systems work? What does this mean for the health and the well-being and disease in this system? Biochemistry is much more about what are the molecular mechanisms which are underpinning these processes. The kind of perspectives are different in that sense.
Brilliant, thank you. Okay, we've had another question saying, is there any research into utilising these pore-forming proteins for combating diseases in medicine?
Yes, there is. The most successful application of these proteins is has been done by a company called Oxford Nanopore Technologies. It wasn't actually this family of proteins, although it's possible they have explored the use of members of this family, and it is possible that in the future they will apply them. Oxford Nanopore Technologies is most famous for having developed DNA sequencing using pores, and the use of this is the reason it's useful is because normally when we do DNA sequencing, we need to have a full laboratory infrastructure. If you're in the field, if you're working in parts of Africa where malaria is endemic, and you want to be sequencing malarial genomes out of people's blood to work out what variants of malaria they've got in them, you haven't got a lab to take the DNA to. So you need to be able to do sequencing in the field. And this amazing DNA sequencing technology, what they've done is they developed something the size of a smartphone, which has a sequencing array in it of these pores, and they can use the electrical current through those pores to work out what the DNA sequence is. It's amazing. It's been developed over the last 20 years. Frankly, when it was being developed, I was aware of it 20, 25 years ago, because nobody really thought it would work. And now it works amazingly. It's very, very successful. And now they can apply it to all sorts of other things. So that is a biomedical development of these pores, because it means that actually you're understanding the genetic basis of disease. Another area where they've been used is that there's a form of tuberculosis vaccine that's been developed where they put in the listeria lysine protein from the listeria bacterium, and they did that because it helps the tuberculosis vaccine to better get into the cells and better cause immunity. So that's another example. Another way is people have thought they might use them to deliver drugs in a targeted way. That's not been so successful yet, but it's still a possibility to use these pore systems to kind of send them, say, to cancer cells with an appropriate kind of tag to target them there, in a molecular tag to target them there, and then to kind of get them to deliver toxins to kill the cancer cells. So people have explored applying these things biomedically, and the DNA sequencing is the most successful one, but there are other things that are possible. And using them as the basis of vaccines is definitely, has always been in people's minds as a possibility and continues to be.
Brilliant, fascinating, thank you. And David, I hope that answers your question, which was, could pore-forming proteins be used to allow for easier drug delivery into cells?
They might well, yeah.
Brilliant. Okay, so if you want to submit your last few questions, please do so now. But I thought I'd just ask, how is the teaching, what is it like to be a student of biochemistry, say in your first year? How are you taught at Oxford? How does it work? What might your week look like?
So in the first year, it's quite structured actually, because what you get is you have your core lectures, and so you will have 2 or 3 of those a day normally. Then on top of that, you'll have your tutorials, and you'll have at least one of those a week, which would be with me or the colleague with whom I look after the biochemists here at Magdalen. That's a key part of kind of developing you and getting used to the university. It enables us to support you as you kind of get into the study skills needed for being here. But then you also have classes that help to support you doing some of the specific kind of areas that you need to study, the what we call the quantitative biochemistry, so the mathematics and the statistics, so the classes in that. The mechanistic biochemistry, so that's some of the organic chemistry you need to know, and the basic protein chemistry, and also the biological chemistry, which is also about the kind of protein and DNA chemistry. So you get classes in that, as well as the tutorials and the lectures and some practicals, and that kind of provides a package which supports you pretty well during the first year to kind of move towards your first year exams. And then in later years, it's slightly less structured, in the sense you don't tend to have the classes, but you do have the practicals, you have the tutorials, you have the lectures. So you're still very closely supported. It's slightly less structured than it is in the first year, because the first year we're just getting you up to speed for being at university, really.
Brilliant. Okay, well, unless anybody would like to submit a last-minute question, I think we will round up there. You'll be receiving an email at some point before the end of the week, I'm hoping, with an evaluation form. It would be super helpful for us as an outreach department if you would fill that out. I've also made a note of some of the suggested reading, so I'll include that in the email as well. If you'd like to ask some students about studying at Oxford, please go and look on the Magdalen website. There's a chat to students function, and I'm pretty sure we've got some biochemists and biologists, etc. on there, and you can ask them about studying here or living here, etc. But yeah, thank you so much, Rob. That was really, really
Thank you. It's my pleasure. My pleasure.
Yeah, thank you very much for coming, and we hope you enjoy the rest of your week. Bye.
Thank you, Amy. Thank you.